here that imagine this is the actin filament, and what these are all active sites. If you imagine, these are all the active sites. Okay, so this is the myosin head. So actomyosin complex. We say this as actomyosin complex, or we call this as cross bridge. The cross bridge. So what is happening with this particular cross bridge that we need to see here? So I'm going to draw that particular active sites. Let me use the same colors that what we use for actin. Then this is the active sites of the actin. Then here I am going to draw that myosin head. So before that, stepwise we'll go with. Now the myosin head is separated, isn't it? Myosin head is separated. Let me duplicate the page that we can understand clearly. Then this is the first step. That means what is required here, what is present? ATP. ATP is present. Okay. Then exposure of the active sites required. That further we discussed already. TNC, calcium bind with the particular. Let me draw that even. Let me draw that even. This is the tropomyosin, and these are the complexes TNT, TNI, and TNC. Wherever the active site present, that all these are going to present here. So the step one, we need to activate this area, and we need to activate this area. We need to activate the thin filament actin. That we need we need to activate the actin, and we need to activate the okay, last screen, yeah. We need to activate this actin, as well as we need to activate this particular myosin site. So how does we get activated the actin? That calcium ions, this is a TNC, this is the TNC. The calcium ion will come and bind and form calcium troponin complex. That will come and form the calcium troponin complex that lift the active sites for the exposure of active site. In the next diagram, I don't draw that particular active site. In the next diagram, I'll show here like this, that active sites are getting exposed. Active site process is over. Okay. What next? That ATP, the calcium ion brings the breakdown of ATP into ADP plus phosphate ions. Okay, so these are also getting activated. Okay, activated myosin head bind with the activated myosin head bind with the actin at the active site that form actomyosin complex. That form the actomyosin complex. Okay, this is the step one. And next, the step two. What is step two? The, at present, what is the condition? ADP plus phosphate ions is the condition here. ADP plus phosphate ions. Okay, so in this particular condition, actomyosin complex is getting formed. Okay, actomyosin complex is formed. Next, what next? Liberation of phosphate ions. Liberation of phosphate ions. So let me draw this. Yeah. Liberation of phosphate ions. Here what is present? ADP plus phosphate ion. Now I am saying the phosphate ion is getting liberated. Then the power stroke, pulling the thin filament towards the H zone. That pulling the thin filament towards the H zone. So what can be happen next? So what can be happen? So these particular thin filament, let me erase the total. Little bit down it will come. Little bit down. The power stroke happened, that the thin filament got down, and our myosin head is okay, so this is the myosin head. Then myosin head need to detach after the power stroke. The myosin head need to detach. How did it get detached over? When a new ATP molecule, ADP ions are getting liberated, a new ATP molecule is attached. Okay. Then again, when this ATP breaks down into ADP plus phosphate ion, then what happens? Actomyosin complex is getting formed. Okay, actomyosin complex is formed. When the phosphate ions are getting liberated, that the power stroke occurs. The power stroke occurs. Okay. Then after ADP is also getting liberated. Then when a new ATP molecule come and attach, that detachment occurs here. Detachment occurs. Then you see that when all these are going, well, when all these things are going to happen, this particular myosin head will face these the one after another, that particular active sites it is going to face pulling the thin filament. Means you need to understand here, see in the picture. So this is the myosin head, pulls, detach, again attach on another active site, pulls, detach, again an active site. Likewise, that myosin head is not moving, just pulling the thin filament towards the head zone. So that means when I pull the thin filament towards the down, my head exposed to the next active site, next active site, next active site, likewise. Okay. So this is the procedure of muscle contraction. When we are doing this particular muscle contraction, so what happens, that events we discussed earlier, and this is a brief explanation. Now, how does the utilization of the energy is going to happen? Okay, for that, let's go with the steps here. Step one, that energy is required. That is so clear. During muscle contraction, energy is required. So the first energy liberate, that utilization, that we know we have the stored ATP. So that we are using that particular stored ATP, we are breaking that ATP in the form of ATPase engine, and that was liberated, that was breakdown in the form of ADP plus phosphate ions, and we said during power stroke, the phosphate ions are getting liberated. Okay, phosphate ions are liberated. Then what next? Can this ADP will be used forever? After the release of one phosphate, this is called adenosine diphosphate. Can we say that we use another phosphate for power stroke? For every power stroke, one phosphate ion is required. Then adenosine monophosphate, adenosine no phosphate. Can we do like this? Can we do like this? No. So that means we need to convert this ADP back again into ATP. That don't say like the liberated phosphate ions are used. So that now the essentiality came here. Whatever the ADP formed here, we need to convert into that particular. This is the step two, usage of muscle phosphagen. For this, let us make a small story here. That, for example, you are running continuously, someone is chasing you. Nowadays, all wild animals are coming on road. So a wild animal is chasing you, a tiger is chasing you, a cheetah is chasing you. Okay, so then what are doing now? You are running now. When you are running, what is your utilization of energy by muscles? That whatever the ATP is present, that ATP you are using, and all that leads to the formation of ADP. Now, the step two, still you are running, that you need to understand that you are running. Okay, so then again and again, still you are running. Okay, so when you are running like this, the second step we are going. The second step, ADP need to convert back into ATP. So for that, we have to use muscle phosphagen. The muscle phosphagen of vertebrates is phosphocreatine. We can say chordate. All chordates, muscle phosphagen is phosphocreatine. If you go for invertebrates, that is phosphoarginine. So what is this phosphocreatine? Which is going to provide phosphate to the ADP. Which is going to provide phosphate to the ADP, is making this again into ATP. So that means one thing you should understand, by ATP form only we can use this particular, using energy for muscle contraction. By ATP form only you can use. So that's why someone came and donated that particular phosphate. Those who donate the phosphate for the ADP to make it ADP back again, that is called muscle phosphagen. The muscle phosphagen of chordates is phosphocreatine. In case of invertebrates, non-chordates, that is phosphoarginine. Okay, whatever it may be, that we got that particular phosphate and we use it again, that ATP will break down into ADP plus phosphate ions, and the certain usage will go on. Usage will go on. We use this continuously. Okay, then as a period came here, all the phosphate is also over from here. Because you are running. So just imagine till I complete this, you are running continuously. So when you are running, so you can see this. It's a first utilization. This is the second one, that what we are using, that phosphate creatinine provided phosphate. So that totally it is over. So now what next? What next? We enter into the step 3. Then now you may feel your breath rate is increased. That means we need oxygen. We need oxygen for what? To break the glucose, to get the energy. Energy is required. You may have that 6CO2 and 12H2O. They may get formed here, but we require ultimate energy because we are running continuously. We came into the third step. Means everyone you can feel this actually. When you run, that initial running with 1 or 2 minutes of that depends here. For some 1 or 2 minutes, some for 30 seconds itself. That I will discuss in the end. Okay, so what happens? We use the first source of energy, then we take the help of the muscle phosphagen. Now that all that particular ATP is over, that's why we are now moving towards that requesting the breathing procedure. That means our nervous system will take care of all this. Then increasing breathing rate, utilization of more oxygen for the more breakdown of glucose for the more energy. Okay, so 38 ADPs are released that you are using now. But we reach a situation, you may have, we are already discussed the respiratory system and circulatory system. That means for supply of oxygen, heartbeat is required or not? That means heartbeat will increase, pumping will increase. But a condition will come here that your demand doesn't equal the supply. These are supplying okay, but you are running very faster, that you are using the energy sources very faster, will reach a particular source called oxygen depth condition. What is oxygen depth? That demand is not fulfilled by supply. Means here supply is insufficient. We say what supply? Oxygen supply. Okay, so that oxygen supply is insufficient. The demands are not going to be fulfilled over. So at the time, do we have any source? Yeah. Is there any possibility without oxygen? Without oxygen, any possibility of little formation of energy? Yes, we say that as anaerobic respiration. Anaerobic respiration. You may have learned over in botany, but I'll give a brief. You may know that there are three procedures occurs in the respiration: glycolysis, then Krebs cycle, then electron transport. There are three steps in the respiration: glycolysis, Krebs cycle, and electron transport. In these three procedures, the Krebs cycle and electron transport require this oxygen, and that release large amount of energy. Okay, that is the advantage. But Krebs cycle and electron transport require oxygen. Glycolysis doesn't require oxygen. Okay, so only glycolysis occurs. What do you say? That is anaerobic respiration. I'm giving in my context, in duology context, that particular issue I'm giving here. Only glycolysis, anaerobic respiration, because there is no need of oxygen. That's why at least it can be happened over. Okay, so here the glycogenesis occurs. That means conversion of the glycogen into glucose, or gluconeogenesis occurs. Means formation of glucose from other than carbohydrates. We are ready with glucose by glycogenesis or gluconeogenesis. We formed the glucose because we need two sources, isn't it? One is glucose, the other is oxygen. Oxygen is not available, at least glucose is available by glycogenesis, breakdown of the glycogen into glucose, and formation of glucose other than carbohydrate. That means from the fats, if it all happens. So, do you have oxygen to break this? No. We have oxygen depth condition, but still you are running. You can't say this total procedure to the tiger who is chasing you. You have to run. That means my oxygen levels are reached low, my demand is increased. That you can't explain that. So that's why our body is going in the step 4 format: utilization of formation of energy, ATP. The process called anaerobic respiration, where it means only glycolysis occurs. Only glycolysis occurs. Do you know that end product of this particular, let me write here only, end product. Generally, glycolysis end product will be pyruvic acid, isn't it? This pyruvic acid is turned into, the pyruvic acid will turn into lactic acid. Lactic acid. This pyruvic acid is turned into lactic acid. Okay. So how you're feeling? In every step you can see how you're feeling. That earlier when you're running, you use the ATP first. Then after the muscle phosphagen utilization, that who provided phosphate. Then after your breath rate increase. Here onwards you can see external appearances, external feeling. You can see that your breath rate, you're observing your breath rate is increased. And then have you feel that, do you feel you're paining? Yes, you're paining. In the muscles, there is a pain. When you're running, your legs pain. That is due to the accumulation of more lactic acid. Okay. So now let us remember what we learned in first year. Skeletal muscle work fast but undergo fatigue. Skeletal muscle work fast, but they undergo fatigue, isn't it? So that feature we should come in now. The skeletal muscle work fast, undergo fatigue. Now we are giving technical information. When they are working fast, we use ATP, we use muscle phosphagen, we use oxygen. When oxygen demand increase, without oxygen, we'll try to produce the energy, and in that case, the pyruvic acid is formed, then the lactic acid, then what? Then what? There is no step after this. That means simply we say fatigue. That means you'll be collapsed over by accumulation of the lactic acid. So we should take rest now. We should take rest now. Okay, so that's it that we have to talk about. The lactic acid, which is formed here. Lactic acid, which is formed here. We should go with that particular lactic acid. Now you just sit somewhere else. You're paining still. Why you're paining? You'll have the answer. Why you're paining? The lactic acid depositions in the muscles. That's why I'm paining. Okay. So then next, what should happen? Where do you see this particular lactic acid? Where do you see? This particular lactic acid is seen in the muscles, isn't it? So now we are moving with the lactic acid cycle. This particular lactic acid, where it is now, it is in the muscles. Okay. So now we need to use our body fluid circulation, heart knowledge here. So from the muscle, we should bring out lactic acid, isn't it? Who collects the blood from the body parts? Superior vena cava and inferior vena cava. So I am taking inferior vena cava. Why? Because that this lactic acid, because of your running, it is found in the muscles, isn't it? So this particular inferior vena cava collects that. Where it goes? It goes to the right atria, then to the right ventricle, then to the right ventricle to pulmonary aorta. Pulmonary aorta to that we reach to lungs. Okay, then from the lungs, get back, pulmonary vein, left atria. I'm going little bit faster here because we learned this already. Left atria, then left ventricle, then left systemic aorta, where we have to go actually. That this lactic acid we need to transport towards the liver. Left systemic aorta, we reach to the liver. Okay, that means left systemic aorta, we use it to say that supplies blood to the body parts. If I want, it will continue. Left systemic aorta, dorsal aorta, hepatic artery, we reach to the liver. Okay, we reach to the liver. So what happens with this lactic acid in the liver? This particular lactic acid in the liver will be converted into glycogen. 20% of it going to be breakdown, and 80% of lactic acid will be converted in the form of glycogen again. So now what is formed? The glycogen is formed. So where do you see this now? In liver. Okay. So that means simply I am saying here lactic acid in the liver is converted into glycogen. Okay. We converted this into glycogen. Then means removal of lactic acid, you feel start relief in that particular area. That means as long as you are removing the lactic acid, that you will find the relief. Okay. So next, so this particular glycogen, where we are now in liver. So who collects the blood from the liver? Hepatic vein. Now I get started again. I have to move to the muscles then now. Hepatic vein. Then again, inferior vena cava. Inferior vena cava to that heart again I reach. Okay, so I am not writing the total story here. Okay, writing in brief. Let me write it. That right atria, then to the right ventricle, then again same right ventricle to pulmonary aorta will reach to the lungs. Okay, from the lungs, pulmonary vein, left atria, then left ventricle, left systemic aorta, we reach to the body parts. So just you need to understand this kind of rotations. We did already in the introduction classes. So that means every time you have to walk through these, every time you need to swim through these. Okay. So that I'll separate this. I'll separate this for you. I'll separate this. So fatigue is over now. That we are not feeling fatigue now. That because the lactic acid is removed and reached to the liver, there it was converted into glycogen. It was converted into glycogen. So what we need to do, what you need to do, then the step one, we need to bring this particular lactic acid from muscles to liver. So you need to use that particular transport vehicles. Now I'm saying I'm not writing. From muscles, you just practice this. From muscles to liver. Lactic acid from muscles to liver. Inferior vena cava. So I am particularly using because we run actually. That means the muscles of the legs we are talking about. Inferior vena cava is going to collect that. Inferior vena cava, right atria, right ventricle, pulmonary aorta. Then we reach to the lungs, pulmonary vein, left atria, left ventricle, left systemic aorta, dorsal aorta, and then hepatic artery, lineogastric artery, that hepatic artery, that no need, just right away we will go hepatic artery, we reach to the liver. Okay, what happens in the liver? We said in the liver that in the liver, this particular lactic acid is converted into glycogen. Okay, so the glycogen or glucose, they get converted. Okay, so what happens after this conversion? Glycogen is formed. Okay, what next? What next? That this glycogen, you need to bring it out. So glycogen is there in the liver. So from liver, you should bring it back to liver to muscles again. So how does it happen? Hepatic vein. Please do remember, not hepatic portal vein, hepatic vein, postcaval vein, right atria, right ventricle, pulmonary aorta, lungs, pulmonary vein, left atria, left ventricle, left systemic aorta, the certain body part, dorsal aorta. We need to say femoral artery, sciatic artery, obturator artery. That no need all these names here. We are going to get all these in medicine. So these particular legs, to the legs, left systemic aorta supplied this glycogen to the left. So we call this particular total phenomena as Cori and Cori cycle, the name of the scientist. Okay, Cori and Cori cycle. So with these, that total story is completed. So let me show you that particular cycle to you, that Cori cycle, that I'll show that particular story of Cori cycle to you. We'll go to the desktop. A picture is waiting for you. Where is it? Cori. Yeah. Yeah, this picture is waiting for you. I need to zoom this. How? Yes. Can you see this? A text diagram. Just text diagram only. This is the story which they are showing to you. That glucose, that 2 lactic acid from here, from the down, we need to start here. The lactic acid, the 2 lactate is converted into 2 pyruvates. And 2 pyruvates are converted into glucose. Where this glucose will reach? Glucose will reach to the particular muscles again, that go to the muscles, that particular glucose in the process glycolysis, that will be converted into 2 pyruvates. That pyruvate will convert into lactose. The 2 lactose will find the 2 lactose into 2 lactate. Again, this is a cycle actually. The 2 lactates will be converted into pyruvates, lactate into pyruvate into glucose. So that particular glucose in the glycolysis, again it forms pyruvic acid. That pyruvic acid will be converted into lactic acid. This lactic acid again converted into pyruvate, then glucose. So then you can understand where what is happening. Conversion of the glucose into lactic acid is done in muscles, isn't it? Then this particular lactic acid is reconverted into glucose in case of liver. So this is the cycle, that interaction between the muscles as well as that particular liver. So that liver, muscle and liver, they'll take care of all this. What do you call? It's a Cori cycle. This is called Cori cycle. Okay. So then